broadly speaking, there are three different ways that you could change the glycosylation. So as we mentioned, changing the post-translational modifications of proteins such as glycosylation influences the structure and the function. So it's very desirable to have certain features, for example, enhanced ADCC or CDC, or change the half-life. So to that end, what we can do is we can glycoengineer the protein or your product to have your desirable attributes.
Hey, smart biotech scientists, welcome back. We are in the middle of a conversation with Róisín O'Flaherty, who is an assistant professor in the Department of Chemistry at Maynooth University in Ireland. And if you haven't listened to part 1 about glycoanalytics yet, please go back. And today we are continuing our journey and we'll be talking about glycoengineering. So stay tuned for an insightful conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Switching our focus to chemoenzymatic glycoengineering, could you clarify what this technique involves and how it distinguishes itself from other glycan modification methods?
So chemoenzymatic approaches involve using enzymes or cocktails of enzymes to change the glycan landscape of your product. And broadly speaking, there are three different ways that you could change the glycosylation. So as we mentioned, changing the post-translational modifications of proteins such as glycosylation influences the structure and the function. So it's very desirable to have certain features, for example, enhanced ADCC or CDC, or change the half-life. So to that end, what we can do is we can glycoengineer the protein or your product to have your desirable attributes. And you can do that in three ways. So the first way is genetic engineering, and that's where you're knocking out certain parts of your molecule that are responsible, for example, galactosylation or sialylation. The second way is metabolomic changes. So you're actually in your cell culture, you're changing the conditions and that can influence the glycans that you see downstream. And then the third way is the chemoenzymatic approach, which is it's fairly rare, I would say. It's not exploited that heavily, but this is where we use enzymes to actually change the glycan landscape. So it's very much done at the end of your process. So it's very much after you have created your protein product, which is heterogeneous in nature in terms of its post-translational modifications. What we then do is we come in with our enzymes and our substrates and we can engineer the conditions that you want. And that's, in my opinion, very desirable, for example, R&D labs, where you're not quite sure what glycan features you actually want. So here you might produce your product and you might do some chemoenzymatic reactions and then test the efficacy of those variants. And if you then know what features you're going after, then probably naturally, I'd say companies generally will go and do the genetic modifications thereafter. But that's where I think it's incredibly powerful. Or, for example, if you have something going out of spec, if you're in industry, you could, in theory, change the glycosylation to bring it back into spec. So that's another thing that you could do if you're doing biosimilars or things like that. There's a lot of scope for this technique to be used across the industry.
Can you provide a few examples of how this technology has contributed to the development of therapeutics, vaccines, or even diagnostics, as you mentioned?
I think probably the most prominent example, for one, it's difficult to know for sure because a lot of this is done in-house by the biopharma companies and they don't necessarily share this information, but you can definitely, I know, having talked to many of these industries, at some of the biopharma events, it's definitely been done, but it's been done at the R&D level. But one big player, for example, is Roche. They are very interested in the chemoenzymatic portion and they have patents and things on this technology. But it's also, I've seen it being adapted by the industry in other places. For example, Biopharmaceuticals, they're in clinical trials in stage 2, where they're using enzyme antibody fusion. So they're basically using the chemoenzymatic approach fused to an antibody. And I think they're in phase 2 of using this technology where they're clipping off sialic acids on cancer cells for anti-cancer treatment. So I'm sure there are an awful lot other examples out there, but it is already being exploited, this technology. And definitely in terms of diagnostics and definitely as a complementary tool for the pharmaceutical giants as well.
This certainly opens up many possibilities for engineering our therapeutics. Now, let's discuss the challenges. Although it's a fantastic technology, like all technologies, it has drawbacks. What are the limitations of this technology?
So the biggest one is cost, I would say, because traditionally the enzymes are pretty expensive. So even the term chemoenzymatic design, that can be interpreted differently for different people. Some people, what they do is they engineer endoglycosidase mutants and then they make sugar donors. So if you're using this technique, then that requires a little bit of chemistry, I suppose, to be able to do those reactions. But I think the biggest hurdle is probably cost because these enzymes are pretty costly. However, if there are certain things that we're developing and other groups are developing where you're tethering these enzymes to solid supports, that significantly reduces your costs because you can reuse your enzymes. The other thing is you can't always be as selective as you might like to be. So most of these enzymes, historically, how they came about was they were used when we were trying to characterize glycans. So we used, for example, a bovine kidney fucosidase when we were trying to clip off fucose residues. And we did that on a released glycan level, which is relatively easy because the fucose residues are easily accessible. However, on a protein level, they're far more protected to these glycan motifs. So we're not always able to selectively remove the glycan motif that we might like on the solid support. So that's definitely a challenge as well, that you might not be able to get the exact homogeneity that you might want. So you really need to tweak with your reaction conditions to get the optimized approaches for your protein. And it depends on what protein you have. Like EPO is really glycosylated. It would be much more difficult to engineer EPO than it would be to engineer IgG. So I suppose time and money, those are the biggest downsides.
You're making a good point about the different forms of glycosylation, because if we take an IgG, the glycosylation is simple. But if you have a fusion antibody, or you mentioned EPO, we have very complex glycan structures. So I'm wondering, what are the technical limitations of this glycoengineering method? And are there certain glycans we are not able to tweak? Or is it just a matter of having the right concentration and the right reaction time?
I think there's an element of both. I think we can absolutely play with the reaction conditions and tweak some of the parameters. And there are little tricks that you can do by adding in salts and things like that to increase the enzymatic activity of your enzymes. But also, the other approach is to produce different. So I collaborate with some of the enzyme companies and we can engineer new enzymes that are smaller and that can access some of the spaces that some of the larger enzymes can't. And that's something that we've done. Fucose is one really good example of that because it's the one that most companies are very interested in to create the afucosylated variants. That can be challenging on a protein level. And one way we can circumvent that is we can make a smaller fucosidase enzyme. And that has been done in certain cases. So you can always find a way, but it sometimes takes a little bit of time, but it's definitely possible. And then for your protein or your product, once it's developed and once you've tweaked all those conditions, it's plug and play.
And speaking of advanced technologies like AI and machine learning, do you think they could assist us in designing better molecules?
Yeah, definitely. I mean, we're experimentalists, so we're actually doing the chemoenzymatic engineering, and then we'll be testing using SBR and things like this, the efficiency and the binding efficiency of our proteins. But of course, all these bioinformatic tools also can predict the outcome of some of these reactions. So one example is Reglyco. So that's a software by Elisa Fada. So I can share the links with you afterwards if you like. But what you can do is you can basically upload your protein structure from the protein database or AlphaFold, and we can artificially add on these glycan motifs, and it can predict the three-dimensional conformation of your sugar, which will allow us to predict how efficient it's going to be at binding if you're interested in, for example, FC binding assays or things like that. So 100%, I think the future would be taking these calculated approaches, and then if we can then supplement that with our experimental approach, it's really beautiful, I think, the combined approaches.
So what I'm understanding, Róisín, is that in the future, we will increasingly work in sync with technologies. As scientists, we will develop alongside technology or ChatGPT or any other advancements truly together, right?
We're already doing it. I mean, if you look across everywhere, Adobe, all of these big companies, they've embraced it. They've embraced it for a reason. It's not going away. On an individual level, it is very scary, the prospects that AI has, but it's also very exciting, and I think you can't ignore it.
I completely agree. It isn't going to disappear. We need to adapt because technology won't stop advancing. If we look at history, progress has never halted. It's always moving forward.
Yeah, and I think the most important thing will be to validate. So our roles will be validating the accuracy of the model as opposed to doing it taking 10 years to actually create this variant. That will take seconds. And our role will be very much a deeper thinking, critical analysis role.
Yeah, absolutely. As we are wrapping up, Róisín, what is the most important takeaway from our discussion?
Personally, I think the most important thing is to always learn and always adapt and always look around and problem solve. And together we'll move forward.
That's a great way to conclude. Always learn, always adapt.
And always move forward, never backwards.
Absolutely. Where can people connect with you, Róisín?
So you can reach out to me on LinkedIn or on Twitter. So on Twitter, I'm Irish Glyco Girl. And on LinkedIn, I think I'm just Róisín O'Flaherty.
You will find all these links in the show notes, so please reach out to Róisín and have conversations about glycoanalytics and glycoengineering. And thank you once again, Róisín, for being on the show today and sharing all these amazing insights with us.
Thanks a million.
Hey, smart biotech scientists. Thank you very much for tuning in. What did you think about today's episode? Please tell us by leaving a review. And perhaps you still have questions about glycoanalytics and glycoengineering, and you're wondering how you can use these tools to the fullest potential in your process development workflow. I would like to help you do that because I have worked many years in glycosylation. I did my PhD on it, and then later did a lot of process development activities where we tuned various glycan attributes of molecules. So I'd like to offer you a free assessment to help you get started. And if this is for you, just book a call using the link down in the show notes. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.